as I got pulled deeper and deeper into the treatment of hydrocephalus, it became extremely important to understand the basic mechanics of CSF circulation, and also the production and absorptive capacity of the brain, because these really dovetail beautifully into the treatment algorithms that we still use today, both on the surgical and medical side. And I'll highlight those as I move forward. You'll hear a lot about in your physiology courses and your applied aspects of neuroanatomy, this idea of a barrier within the CNS, and specifically the brain itself. And this barrier one can think about in many ways, from the standpoint of whatever field you might pursue, infectious diseases, anesthesia, psychology within a brain interface. There's a lot to be revealed from the standpoint of this idea of a blood-brain barrier. As we're talking about CSF physiology, production and absorption, really we'll be focusing on some of these elements here. I think of these as really the microanatomy or the macroscopic aspect. That's the cellular basis of this blood-brain barrier. And then the biochemical features, which pharmacology is all over from the standpoint of modulating, bypassing, and tricking the blood-brain barrier into allowing penetrance of certain molecules. And I'll focus specifically and mostly on the epithelium that I'll get into in a little more detail here. In your histology course, if you haven't seen already, the principal building block behind this blood-brain barrier is what's called a tight junction. And this tight junction is really unlike any other capillary interface in the body. Capillaries and parenchymal interfaces are really defined by the idea that things can really percolate quite readily between the plasma and the extracellular, and then ultimately the intracellular matrix. On this EM, this barrier is built around this, what's called zonula occludens, or this tight junction. This is the adluminal part of the capillary membrane here. But it's really an interdigitated aspect of the cellular membrane that, if you think about it, this cell membrane then continues as one holistic membrane across the entire cellular substrate. So whatever behavior this membrane bestows upon the cell itself is bestowed upon that whole tissue plane. So we're going to talk about this as it plays towards the choroid epithelium, as well as the epithelium of the capillary as well. And this becomes extremely important. I use this set of cartoons to drive home this point, where the different chambers, here's the, if you will, the capillary choroid plexus, the CSF in this light blue, the brain parenchyma in blue, and the subarachnoid space in gray. And I'll go through this from the standpoint of where this barrier exists, or where the blood-brain barrier exists. For most purposes that you will be dealing with in your medical school and beyond, it's really this interface between the blood and the brain parenchyma that you will be focused on, as far as that really solid substrate of the blood-brain barrier. And that's depicted here in this bold. We're at the level of the capillary endothelium. As you get down to the nearly cellular size, as far as the luminal size of that blood vessel, that barrier exists between the capillary endothelium and the brain parenchyma. So it doesn't have that quick transmission of plasma substrates that can gain access to the parenchyma. Of interest, that continues as the blood vessel passes through the ventricular compartment. And because of the properties of choroid plexus that we'll talk a lot more extensively about, this is a very nearly fenestrated membrane between the endothelium and the choroid plexus. But now that tight junction exists at the apical area of the choroid plexus epithelium. So instead of at the capillary level, when the blood vessel transgresses the CSF chamber, the ventricle, or the subarachnoid space, that is where that blood-brain barrier exists, on the capillary epithelium, the choroidal epithelium. All of these are off. These highlights are supposed to denote, thanks to my slide formatting, what are called circumventricular organs. So if we focus on this idea that there is an absolute blood-brain barrier, like every rule in medicine, it's meant to be broken. And these were meant to denote certain areas of the brain where this blood-brain barrier doesn't exist. It no longer exists from the standpoint of that absolute interface between plasma and brain parenchyma, or histology. They're listed here, not that you have to know them, but I think if you think about their function in the normal neurophysiology, it makes great sense. Choosing an example such as the area postrema, which is usually located in the caudal aspect of the medullary segment of the brainstem down here in the caudal fourth ventricle. It's also called the emetic center. So things that are exposed in the plasma that are considered to be toxic or somewhat ill-fated gain quick access to the neurons of the area postrema, the emetic center, and cause this reflex of vomiting. Others, the adenohypophysis, or part of the pituitary gland, very, very rich fenestrated capillary network, which helps control water balance very rapidly. You know, the human body cannot tolerate wide swings of osmolality or sodium. So it has this rapid transmission between plasma and neurons to institute changes, and hence the blood-brain barrier is broken down in these areas. And you can usually see this on contrast neuroimaging, usually MR, where with contrast agents, you'll see a rich enhancement in some of these areas, thought to be due to a visualized aspect of the blood-brain barrier that is broken down. As depicted here, you can see this wide gap in between two cells, unlike what I showed you before with the tight junctions. You know, of course, that poses problems to the clinician, and we won't spend a lot of time talking about this, but you know, the brain, unlike other organisms, won't see the types of concentrations we want of certain agents, whether they be antibiotics for warding off infection, chemotherapy for cancer. So, of course, clinicians and basic scientists have challenged themselves for decades and decades in how to overcome this idea. How do you get drugs into the place we want them? I've listed these based on kind of a regional distribution or global distribution. You don't have to trouble yourself with identifying each of those, but suffice it to say that at the interface of pharmacology for any of these purposes, there's a lot of work that goes into manipulating the blood-brain barrier. One of my passions has to do with getting drugs to brain tumors themselves, and anybody who's been around the realm of neurosurgery would know that there are certain biodegradable polymers that you can place on the brain. This is a cartoon depicting that idea that the red blood cells here in the capillary, monoclonal antibodies delivered through the capillary, would never gain penetrance into the parenchyma because of their overall size and this idea of a blood-brain barrier. So other ways to get around there is to place a microcannula outside a blood vessel itself, whether they be viral particles or monoclonal antibodies, whatever the case may be. There are plenty of strategies to overcome this idea.
Hey everyone, Ryan Rad here from neurosurgerytraining.org. If you like that video, subscribe and donate to keep our content available for medical students across the world.